Good morning, everyone. Welcome for this live webinar. We will be starting in a minute. Dr. Divya Ravishankar sir has joined. Good morning, everyone. Welcome for this live webinar. We will be starting in a minute. Dr. Divya Ravishankar sir has joined.
Hi, good morning, Dr. Mujib. Yes, and everyone. Doctor has joined just now.
What is the name of doctor?
Sai Ganga Panesia.
Okay, Sai Ganga Panesia.
Yes.
Okay, give me 1 minute. Okay. So good morning, Ravishankar sir. You are able to unmute now?
Yes, now I'm able to unmute.
Yeah, so I was searching for the name of
Dr. Ravishankar.
Right?
So I joined using our commercial, Zoom license.
Yeah.
Can you all hear me?
Yeah.
Dr. Mujib, yeah, okay, perfect.
Absolutely.
So I was just joining with my company's name.
Yeah.
And
So I have made everyone on mute, and they cannot switch on their videos, and they can just chat with you, so that it avoids your distraction, and it is being live streamed on YouTube also.
Perfect.
People can join here and as well as there. So because you are a co-host, you can talk to me, and if anything is there, Uma Priya and Dr. Divya can talk to us. Do you want me to make anyone of other people's as co-host? Please let me know, Dr. Vin.
Yeah, Dr. Divya is there. I think should be fine.
Yeah.
She'll handle from my side.
Yeah. So without any delay, we'll start this session. And today's agenda would be, we'll be having the introduction for the host organization, which is Clinosol, for 2 minutes. And then introduction about Dr. Ravishankar sir and SGP for 2 minutes. And then we'll start the session without any delay. So I'm sharing my screen, and okay, so about the hosting organization, I'll be taking 2 or 3 minutes. My name is Mujib. I am the founder and CEO for Clinosol Research. I am basically a pharmacologist, and I hold MSc in clinical research also. So our services includes clinical research trainings and consulting, and also GXP trainings, ECPs or GLPs or GMP trainings to pharma companies and CROs or to the trial investigators. So these are our timelines. So I'll just brief in a snapshot that 2021 we had 400 admissions and 390 placements, and we have got a couple of awards, and we published 5 newsletters and quarterly magazines 3 and 270 plus blogs, 50 articles. And in 2022 we have taken around 200 plus admissions and 150 plus placements from different companies. We collaborated with ACDMs, ACRPs, also SCDMs. We have registered our office in Canada also. We started office in Bangalore. We have received 3 awards in this year. We have got collaborated with PV Edge and Clinion database. 150 plus blogs were published by our students, 100 plus presentations were being given. And we are academic partners for SCDM and ISCR conferences, and 40 plus industry expert interactions, including this. We are having every weekend interactions for the benefit of healthcare professionals. So these are the courses: clinical research, CDM, pharmacovigilance, medical writing, regulatory affairs, database programming, project management, TMF, and SaaS and system validation courses. And the duration varies from 4 months to 6 months, and total credit is 200. Eligibility is anyone who is freshers from bachelor degree of medical or allied or pharmacy or life sciences or biomedical engineering. And these are a brief snapshot of the USPs, unique selling points for Clinosol, the best placements and national and international presence, in-house library, 2 years of validity, industry visits, industry collaborations, alumni experts, ACRP, ACDM, SCDMs, and PVHN Clinion. So with this, I am stopping my introduction. I think I know it was very quick because I wanted to hear more from Dr. Ravi Shankar sir about the SGP and PSA. So let's have a small intro of Dr. Ravi Shankar sir. I'll start my sharing and a short video.
Being an allopathy doctor, and that too professional as cardiovascular surgeon who has done 3,000 cardiac surgeries, has magically converted his direction to establish a new science called polyscientific Ayurveda, which is a combination of modern science with ancient Ayurveda. He is none other than Dr. Ravi Shankar Polisetti, founder of Polyscientific Ayurveda, which is driving Ayurveda in modern terminology.
Dr. Ravi Shankar Polisetti is a cardiovascular surgeon, and he is the member of Cardiovascular Association across the world, including the PCSI of India. Because of his humble background, being a student of Bhagavan Sri Satya Sai Baba, he had taken up Ayurveda as a special research of interest and has evolved a new integrative medicine called Polyscientific Ayurveda. And based on his translation research of Ayurveda, he evolved a digital IoT platform for healthcare, which could potentially revolutionize the way healthcare is delivered. Let's listen about PSA from him.
That's a short intro, Dr. Ravi Shankar sir. Thank you so much for sending out that video. So all the people in this call would be requested to post your question in the chat box so that Dr. Divya can make a note of the important questions and can put these questions to Dr. Ravi Shankar sir at the end of session. I'll be asking my team to unmute everyone at the end of session. So without any delay, we'll start today's session. Ravi Shankar sir, you can share your screen. I think,
Yeah, I'm here.
Yeah.
Hey, Dr. Mujib, thank you very much, and wish everyone a wonderful Ayurveda day. Today is, incidentally and coincidentally, it's Ayurveda day. I wish everyone a great day today. And I humbly present that, it's going to be our duty to validate this amazing science of Ayurveda, which I have done in my own way, in our own way in the last 20 years. And I want to present, certain beautiful aspects of how we personalize even prescription of allopathic drugs based on Ayurvedic, translational research. Friends, this is there's a very elaborative course, essentially, not a course. I mean, the lecture itself is a elaborate 545 hours of duration. I'll not talk for the whole 545 hours, but then I'll certainly talk for the prescribed time and take it from here. And I would say Dr. Mujib is a wonderful person, and I met him about a few weeks ago. I mean, not because he's hosting me that I'm saying these good words about, but he's a very dynamic, a very, very great leader. And, his team is a wonderful team. I just met them all, very recently. And we have, signed up an MOU so that we work together in bringing out, certain very advanced research concepts from our own ancient science. Now, we agreed, not just today, but long ago, that if there is science in our ancient science, let's adore and embrace it. If there is no science, I mean, we will discard it. That's been our, unspoken rule. So let me just present you quickly the plan for today's invite. Quickly about polyscientific Ayurveda. Why polyscientific Ayurveda? Coming from a cardiac surgeon, and what are the important aspects of the translational research that we did? Just so that we all know, we were the first in the world to actually model the Vata, the Pitta, and the Kapha, which are the fundamental concepts of Ayurveda. We modeled them in experimental animals and generated data. And based on this data generated, we evolved this concept of polyscientific Ayurveda because we found some real science in it. And I'll share very few aspects of this translational research in the second part, which could last for about 15 minutes. Then I'll show you how the pharma drugs could be personalized, and then of course, I'll also talk about this new digital technology that would help us, really put India ahead by at least 15 years than the United States of America. And then, I'll also, see if we could actually position India as the health capital of the world and future directions. And then I'm all there to take your questions, please. So this is how we're going to, structure today's lecture. So let me just quickly tell about the fundamental tenets of polyscientific Ayurveda. See, what we did was, we modeled Ayurvedic concepts in experimental animals and then generated data and, try to understand the problems that are currently actually afflicting the modern science and then the research and the research fraternity across the world. So with this, what we found was there is real and interesting applications of this translational research, and we were able to translate this entire knowledge generated from translation research to improve clinical outcomes, and we were able to help a lot of end-stage patients, and I'll show you how. So friends, polyscientific Ayurveda is not just about Ayurveda. I would actually say that it's a very advanced form of allopathy, and you will see very quickly why I position it so. These are the several disciplines like that are encumbered in polyscientific Ayurveda. You all know, we all are researchers. I'm assuming, and I'm sure most of you must have come from the background of pharmacology, MD, PharmD, or even PhDs in pharmacology. So some of you must be students and some of you must be researchers already. But then, as you see, there are several disciplines here encumbering data science, applied math, Python as a software programming language, the concepts of lab technician, manufacturing processes. All these concepts actually are bundled in the form of polyscientific Ayurveda. So you would see that this is not just about Ayurveda, but it is about an advanced form of allopathy, and that too coming from India. So friends, let me just introduce, why Ayurveda? Why was I actually looking for Ayurveda is, as you see that the World Health Organization defines health as a state of physical, mental, and social well-being, and not merely the absence of disease or infirmity. Now, for diagnosing a disease, we do run a battery of tests, isn't it? If you talk about, today's topic is on diabetes, so that's why we'll talk about the current problems in modern medicine in respect to diabetes. Generally the problems are that we are able to cure 15% of acute stage diseases. About 45% of the acute diseases are being converted into chronicity, not because we want to, but that's where the modern science is right now. And about 40% of diseases, we don't even know where they come from, like autoimmune disorders. We don't know why the body is producing antibodies against its own tissues. There are several theories running around. There are several diseases like senile dementia, Alzheimer's, Parkinsonism, epilepsy. Talk about several diseases like cancers. We don't know why the oncogenes, which are present in all of us, are not getting expressed. They're getting expressed in only about 1.7% of the population across the world. So, you see there are several problems, several diseases for which the etiology is not known. These are the current problems. Quickly, a synopsis of this. And I'll go a little deep into the debate on blood sugar levels today. Look at what American endocrinologists are saying. They're saying, if you took they're saying that it is safe to maintain your HbA1c below 5.5, and when you're treating a patient, it should be below 6.5, the HbA1c. I guess, I hope you all know the value of the HbA1c, right? The American Diabetic Association is saying that they're okay with maintaining your blood glucose levels, HbA1c at in normal individuals below 6.5. But then in diabetes, you maintain it below 7.5. The American Geriatric Association says it should be between 7 and 7.5. The American College of Physicians is saying it should be between 7 and 8. And again, a trial, a CORD trial, ACC-ORD. This trial mentions that your HbA1c, they found a certain interesting correlation that if the HbA1c is maintained around 8.4, the patients who had concomitant heart disease really did well. And when they targeted aggressive glucose lowering therapy and maintained it between below 6.5, as suggested by the American Association of Clinical Endocrinologists, 33% of them had a heart attack, and out of which at least 50% of them had fatal heart attacks. And American Association of Veterans, or American Veterans Diabetic Association is saying that their patients are doing well with 9.5 HbA1c. So friends, the point that I'm trying to drive is several organizations are giving you different HbA1c values. And the problem with the family physicians or diabetologists across the world is, which standards do you follow? Isn't it a big question? Which standards do we follow? We don't know. Is it good to maintain below 5.5, or is it good to maintain around 9.5? We don't know. And mind you, all of these associations are data driven. They're not just, they're not just just like WhatsApp groups. Several WhatsApp groups have messed around the psyche of Indians and across, not just Indians, but people across the world. People are really confused. Why? Because WhatsApp groups, perpetuate lies. Not lies, they could perpetuate anything that comes into their brain. But these associations I have immense respect for are all data driven. All right. They all are data driven, and the data from American endocrinologists is saying that the HbA1c should be below 5.5 for normal individuals and below 6.5 for a diabetic individual, and then so on and so forth. As we mentioned, there's a diversity in terms of what values do you maintain. Isn't it a big question? No, the debate not just on sugars, this is really led to a big debate among Indian doctors. We don't know, in 2019, when American College of Physicians said that your HbA1c should be maintained between 7 and 8, while we were all targeting to maintain it below 5.5 or at least below 6.5 in diabetic patients. The American College of Physicians recommendations came as a big jolt. We really don't know what to do now. And so we are questioning those norms, because, most of the guidelines are coming from the US, we're not, from these associations across. I'll take the questions a little later, please. Please keep posting these questions in the chat box. So the question is, how do we standardize? Not only this, the debate on cholesterol is also open. We're not going to into the nitty-gritties of cholesterol debate, but then I would certainly make it a point that based on the data that is available today, even high HDL is causing heart attacks, friends. Isn't it a news? This was published in 2019 in a certain cohort of 7,000 plus patients. In 95% of these 7,000 plus patients who had high HDL suffered a heart attack. And when the biopsies were done of the plaque, they found about 95% of them had high HDL, high HDL in the blood and also HDL in the plaque itself. Isn't it a news for us? Now, low HDL is anyhow associated with heart attacks. We know that. And we heard that high LDL is causing heart attacks. That we know already. But did you hear that there was a researcher called Malinsky et al., an entire Danish research group? This is a German research group that retro-analyzed about 122,000 patients, out of which 75% of these patients who had heart attacks. You know why they had a heart attack? According to this group, they had a heart attack because they maintained their blood lipids, LDL and triglycerides levels below 100, which is actually prescribed, which is what we are, AHA, American Heart Association, is prescribing to maintain LDLs below 100 and also TGLs, triglycerides below 100. And this group, Malinsky et al., with a huge, 125,000 patients, is a big cohort, friend, out of which 75% of them had heart attacks because of low LDL and low triglycerides. This is not where I want to land. I want to start the debate with diabetes and then see if we could actually solve it from India. The dilemma is pretty understandable because, Human Genome Project itself started off this dilemma. I'll just quickly pose this dilemma again, to you all, so that we know where this confusion is coming from. One of the views of where this confusion could have been coming from is that a Human Genome Project, which promised literally answers for all the questions, ended up saying that there are 3 million genes in the genome and only 22,000 of which are codons. We know that the rest of them could be histones and several other, structures like microRNAs and all. But then only 22,000 codons present, which means based on the Crick and Watson model, there could be only 22,000 proteins. And we really don't know how did 2 million proteins come into the blood, into the body from just 22,000 codons. And of course, there are several explanations coming now, from the field of the microRNAs. There are transcriptomics, there are translationomics, the metabolomics, proteomics. There are several omics sciences coming in to explain this versatility of how so many proteins evolved from just 22,000 codons, friend. That's why we have epigenetics now, and then there's several sciences trying to explain this. But we have a problem. The basic problem is that we know only 1% of the genome as of now. 22,000 codons, we know what they're doing, but the rest of the 99.1 or 3% of the genome, we don't know what it is doing. We are still figuring it out, number one. Even if we know, okay, we say, do we know this 22,000 codons thoroughly? I'm taking a quick assumption from, Nobel and Peach transactions in computational systems biology. If two genes end up producing a certain physiological function, then you know what? How many functions do we end up with in the body? Physiological functions, 450 million. And in guidance physiology, we know that the body has only about 80,000 functions, friends. More than 800,000 reactions are documented, and so on and so forth. So we know only 1% of the genome. We know less than 0.0007% of the physiology, potential physiology, and even with such minimal knowledge, we're able to cure 15% of diseases. Isn't it great? But can it be better? Is there a better way of doing it? This is the question that led me into understanding Ayurveda, trying to understand Ayurveda. So, with my, background, being taught Sanskrit very early in my age, and I started reading these books, friends. I stumbled upon a beautiful verse. This is really great. Why? Because today we don't know, with this dilemma that I presented, we don't know which blood glucose value is good for which individual. Because veterans are doing good at 9.2, the certain athletes are doing good at 8.4, and certain researchers are doing good with their blood glucose levels at 8 and so on and so forth. Which is good for which individual, we don't know. American College of Physicians again is saying that you need to personalize the blood glucose level goals in your patients. And Ayurveda, look at this definition. It says that there is something called samadoshaha, samagnishtha samadhatu malakriya, prasannatme manasendriyaha swastya iti. What it means is that there are three agents, samadosha. The concept is revolving around certain doshas. Many of you must have heard, certainly. But what the ancient science is saying is, if the three doshas, which are the vata, the pitta, and the kapha, are in equal proportions in each of the 7 tissues and 42 organs, then swasthya means health. Again, there's an elaborate explanation for this word swasthya. I don't want to go too much into the Sanskritology, but then, let me help you understand that if these three doshas are present in equal proportion in each of the 7 tissues and 42 organs, then the body automatically restores its health, is what this shloka is saying. So how do I translate that into the language of our medicine? Our understanding is that when these three doshas are in equal proportion in each of the 7 tissues and 42 organs, what happens is that there is a homeostasis at genetic level, proteomic level, subcellular pathway, the level of the cellular level, tissue, organ, and organism level, and leads to physical, emotional, social, mental, and spiritual health. This is what this ancient shloka is saying. And then I started dwelling deep into these concepts of the three doshas. What are they? When I started off, I stumbled upon these properties attributed to each of these doshas. The three doshas are attributed by this dosha. The properties, vata is attributed by shitala, vishada, ruksha, khara, which means the cold, bright, dry, rough, hard. These are like very vague. That's what at least I felt, at the time when I was reading these books. I felt this is a very vague presentation of the body, when we were taught that the human body is comprised of 7 million genes. How could these three doshas, which are again, there are no chemical composition here, they're just properties, certain properties mentioned. We really don't know what they were doing. And, it really led me to a bigger dilemma of, whether this science was really valid or not. The pitta is again attributed by seven of these properties. Kapha is attributed by 7 of these properties, 7 to 8 of these properties. But then, interesting friends, what I stumbled upon is really interesting, because this is from the Puranas, but I think I should post the reference from Rasendra Sarasangraha. The book is right here in my cupboard. The green book that I'm pointing out and the red one above that are those books from where these references are taken. What they mean will baffle you, friends. What this shloka is referred to as Ashitischatva Laksha Rasamritam Swastya Iti Abhidiyate is what is saying. What is one of these books is saying. What it means is Ashitischatva Laksha Rasamritam. Ashitischatva Laksha means 84 lakhs. Rasamritam, nectarine chemicals, are maintaining your health. That really, humbled me. I said, we were taught that the body is comprised of 7 million molecules, and then the Asian science is already talking about 8.4 million molecules. Then I went to my professor in biochemistry, who was a Nobel laureate, Professor Chirkin from Belarus. He said, yes, Ravi, I would say, when I was 19-year-old, 20-year-old at that time, he said, Ravi, there are 12 to 20 new molecules being found every day in the human body. I said, sir, this ancient book refers to this. I said, yes, I would believe that, it is so, because we are discovering 12 to 20 new molecules every day. And that was in the year 1993. And then, in the next 270 years from 1993, I calculated that we'll figure out the rest of the 1.4 million chemicals. It would take 270 years for us to figure out. Not only that, friends, we all know the value of solubility, isn't it? M.Pharma students or PhD students who have done some work in analytical chemistry would agree with me that solubility of chemicals is very important for us to isolate them, separate them, then, subject to XRD, and then, structure elucidate these chemicals. You know what? Interestingly, all of the 8.4 million chemicals present in the body, as mentioned in this text, they're already profiled based on their solubility. Jalaja Navalakshani, 9 lakh chemicals are water soluble. Sthavara Vimshati Lakshani, 20 lakh chemicals are stable compounds, non-reactive. Guruta Chaturasi Lakshani, 4 lakh chemicals are, fat soluble, and so on and so forth. 16 solvents are mentioned that would help these 8.4 million compounds to be, made soluble, and then you could isolate. Once they are soluble, you could isolate them, then structure elucidate them. I found this is amazing. And then I went further reading them. And then I found that the 8.4 million chemicals in this ancient text, as mentioned in the ancient text, participate in about Trin Shannama Sahasra Kotyacha Rasayana Karshani. What it means is 32 trillion chemical interactions are possible. They're not talking, they're very specific about clearly mentioning whether it is a chemical interaction or a chemical reaction. There are specific words used. Chemical reaction is rasayana charya. Chemical interaction in the ancient text is mentioned as rasayana karsha. 32 trillion interactions possible among these 8.4 million chemicals, friends, and they classified them into 21 broad categories based on their properties. The properties of these interactions, like for example, when we say van der Waals forces of attraction, you mean that the forces of attraction of, say, nucleus of one atom with the electron cloud of the adjacent atom, is all classified under van der Waals forces of attraction, and so on and so forth. Our ancient science already mentions about 21 broad categories of such intermolecular, intramolecular forces, and they classified them into 21 broad categories. 7 of these categories drive molecular pathways, 7 of them drive metabolic, and the rest of the 7 drive assimilative pathways. And biochemically, they were classified as Vata, Pitta, and Kapha, friends. Very summarizing the entire biochemistry that I learned, that I heard from these ancient texts. It looked very astounding for me because, I never thought that, biochemistry would even be represented in our ancient classical literature. And then what does a quick summary of what is happening is how they presented this science to us is that everything, all the physiological pathways, the immunological pathways, the biochemical pathways in the body were classified based on their Vata, Pitta, and Kapha fingerprint. And not only there's the foods that you eat, the moves that you make, the thoughts that you make, the moves, the physical moves that you make are already classified. Even the exercises that you do, the yogasanas, the Bhastrika, for example, it says that the Bhastrika aggravates Pitta. For example, the Pushya Nakshatram today, tomorrow, the Pushya Nakshatram, the star, aggravates Pitta. That's how precisely they classified everything that you see around has a certain Vata, Pitta, Kapha fingerprint. And what you do is essentially, understand the Vata, Pitta, Kapha fingerprint of an individual based on a certain technique. There are several techniques. There are more than 8. Some mention Ashtavida Pariksha, some mention Dashavida Pariksha, which means there are 8 methods of investigating the human body, and there are 10 methods of investigating the human body. In all, the urine analysis, the stool examination, the blood examination, the skin examination, the pupil, the tongue, the skin, and the pulse. Based on these several types of methods of examination, you essentially understand the Vata, Pitta, Kapha fingerprint of an individual. The Vata, Pitta, Kapha fingerprint of an individual involves exact measurement of the proportions of these 3 agents of Vata, Pitta, Kapha in individual, about 42 individual organs in the body, friends. This is how you diagnose the patient through Nadi Pariksha, through pulse diagnosis, and by feeding, by giving appropriate diet. Appropriate diet means you give a certain individual that foods, you select those foods that bring about a VPK balance in individual organs and tissues in that particular individual. So you cannot just randomly choose a food, say your keto diet is going to treat your diabetes, or your millets diet is going to reduce your blood glucose. That's not what is prescribed in Ayurveda. Ayurveda goes very deep. helps, I mean, from what I understood, it helps us personalize your diet based on your own Vata Pitta Kapha fingerprint. So each person has a unique VPK fingerprint, and that if you diagnose, if you're able to exactly estimate the proportions of these three agents called Vata Pitta Kapha, then what happens, the body, and then prescribe appropriate foods, appropriate exercises, appropriate lifestyle modifications, appropriate herbal supplements in their appropriate formulations and their extraction process. Then when you do this, you bring about a VPK balance in individual organs and tissues of a certain individual, and the body automatically restores health. This is quickly about an intro of Ayurveda, friends. I mean, from what I understood, but theory looked exceptionally great. I mean, at least from what I heard, what I read from the scriptures. But is it? How do you prove it? Was the next question that I had. So let's get into translational research very quickly. So, because the key here is that these three agents, the agents of Vata, Pitta, and Kapha, should be in equal proportion in each of the 7 tissues and 42 organs. What I did was, we tried to create models of Vata, Pitta, and Kapha in experimental animals, and try to see if there were any correlations to whatever is measurable. So what we did was, the guidance physiology starts with action potential. So it was very clear for me that if you see, I'm talking to you, you're seeing me possibly on the screen. Why? Because the light that is coming from your screen, LED or LCD screen, is incident on the retina. In the retina, this light that is incident from the screen is translated into an electrochemical message in the rods and the cones. This electrochemical message is packaged in the form of an action potential. Then the action potential goes all the way to the brain centers in the occipital. I mean, you have the visual center in the occipital region. There, the brain analyzes the signal received, processes it, and then it gives an appropriate response. Even that response is in the form of a electrical activity. So to cut the whole story short, what the body is doing is converting the external signal that is coming in the form of any signal in the through the sense organs, translating it to an electrical current, electrochemical message, which is called as an action potential, and then transmits it to the brain. The brain analyzes this electrical information, processes it, and sends responses to efferent end organs. So what we thought was, if the Vata, Pitta, Kapha were real, then they should have some impact. So how do we create models? What we did was we fed 3 different groups of animals with Vata, Pitta, and Kapha aggravated diets. So these diets were chosen from the ancient texts, those diets that would aggravate Vata, and we fed those diets to one of the groups. The second group was fed with Pitta aggravated diets, and the third group was fed with Kapha aggravated. And then 2 groups were control group of rats, which were fed on balanced diet, friends. And what we found, and what we did, based on applying, there are several principles that are guiding the dosha ergonomics. So what we followed those principles, which are not quite really, it is not in the scope of this particular lecture, because as I said, it's a very exhaustive lecture, friends, 545 hours. I'm trying to give a glimpse into that, kind of a teaser into that big course later. So we created these models by feeding these rats with the appropriate diets for over a month. And after a month, the rats were decapitated, and based on a certain Langendorf method, we isolated the hearts and then, measured action potentials. We measured the LVEDP and, several parameters that we measured. And then I'll quickly, and then how did we measure? We measured using optical mapping system, and glass electrodes were used to actually reconfirm the measurements that were received from optical mapping system. And a quick recap, as I said, what happens when an external current is incident, when the resting membrane potential is around -90, friends. So you all know that when an external stimulus excites this neuron, what happens is slow sodium channels open, sodium ions move into the cell slowly until they reach a certain threshold. The graph is slightly, acute, more acute. It is less than 45 approximately. But then there's a sudden steep acute increase in the slope of this particular graph, because at the threshold membrane potential, which is around -65 millivolts, fast sodium channels open up, and then there's a quick influx of sodium into the cell, and then there's a spike. Once it reaches once it reaches 0 to +5 in different tissues, chloride ions move into the cells along with sodium so as to neutralize the positive effect caused by sodium influx into the cell. And then potassium ions at +20 move outside the cell, pushes the potassium ions outside into the ECF. And then, the graph turns down at +35 to +20 to +35 in different tissues. And then, at the end of this action potential, and then this is how the action potential is generated, friends. And this is packaged into an electrochemical message, as I mentioned, which is called as action potential. The action potential travels all the way to the brain. And then what happens at the end of the action potential, once the brain receives this signal, the brain said, okay, the brain senses it. And then once it receives, then what happens? The cell comes back, brings back its ionic homeostasis. What does it mean? Sodium is pushed out of the cell, potassium is brought into the cell, chloride is pushed out of the cell, and this is an energy-dependent process. Sodium potassium ATPase uses up ATP to and pushes the sodium channels. 3 sodium ions are exchanged for 2 potassium ions through sodium potassium channels, friends, and the original resting state is restored. What I found was really interesting, friends, that the action potential is in different, as we recorded on the optic mapping system, the action potentials are different in Vata, Pitta, and Kapha in their structure. What was different, friends, I summarized in this table. The threshold membrane potential, at which potential the sodium ions, the fast sodium ions open up, channels open up, was around -65 in all of the tissues. But in the Vata aggravated diet-fed rats, what happened was that resting membrane potential was around -72. In the Pitta, it was -90, and in the Kapha aggravated diet-fed rats, it was -108, friends. What it means is the difference. Now let's go to the next table. Look at the third row. The difference between the resting and the threshold membrane potential in control group of rats, which were fed with normal balanced diet as prescribed by the Russian Institute of Nutrition, had 19 millivolts of difference between the resting and the threshold membrane potential. Number one. The Vata diet-fed rats had only a difference of 7 millivolts. The Pitta Pitta's aggravated rats had 25, and Kapha aggravated rats had, the hearts had 43 millivolts of difference between the resting and the threshold membrane potentials. What it means is that the Vata tissues would require a stimulus which is equivalent to as less as 7 millivolts to excite or generate an action potential, and the Kapha would require 43 millivolts of action potential equivalent stimulus to generate an action potential, friends. This is very, 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 very important. Why? Giving a very absurd example, but then this just for mathematical approximation and understanding, I just I'm assuming, if you give an external stimulus, look at the fifth row, friends. When you give an external stimulus of 53 millivolts, what happens is the control group of rats, generate only 2 action potentials. The Vata aggravated hearts generate 7 action potentials. The Pitta generate 2. Kapha generates only 1. And when the external stimulus is only 7 millivolts, none of them but for the Vata aggravated hearts produce action potentials, because any action potential, any external stimulus, which is lesser than the difference between the resting and the threshold membrane potential, is not carried forward in the form of an action potential. The action potential doesn't get generated. That's why you need this external stimulus should help the membrane potential to cross the threshold only when it gets packaged in the form of an action potential, friends. So what you summarize from this is that the Vata tissues are hyperexcitable, the Kapha tissues are hypoexcitable electrophysiologically. So what is happening is Vata tissues are using up more ATP All right. So their demand for ATP is higher, friends. Kapha tissues, their demand for ATP is lesser because they're hypoexcitable. They're not generating action potential. When they're not generating action potentials, then there is no way of using up ATP faster. Or even, I mean, they're lesser there. They're using up ATP lesser than the control group of rats, which were fed in balanced diet, and they were hypoexcitable, friends. This is really interesting. Not only, it is interesting, but what we did was, I showed you, in one of the slides that there are 7 properties attributed to Vata. One of the properties attributed to Vata was Chalatva, friends. And Chalatva, applying Sanskritology, because Sanskrit is a very advanced language, friends. We have to accept it because, this language is guided by 9 sets of grammar principles. Say, for example, the language that we are speaking now, English, has Renan Martin. If someone has read, we all must have read the Renan Martin grammar books. And what it says is, there are over 900 to 950 grammar principles that are guiding, this English language. Tamil language is guided by Tolkappiyam, with about 1300 grammar principles. Telugu grammar is guided by the Chando Vyakarana Sutras, which are 1600. The Hindi by modified Panini Sutras and about 1700 grammar principles. But in all, what? The Sanskrit language is guided by about 200,000 grammar principles, friends. And these grammar principles are classified into 9 different libraries, and hence this bhasha or language is called as the Nava Vyakarana Bhasha. Nava is 9, Vyakarana is the principles of grammar. Now 9 sets of Vyakaranas are guiding this language, which means if you take one matra, one syllable in Sanskritam, you apply at least one principle from each of these 9 grammar principles, you end up with 9 meanings for the same alphabet, friend, for the same syllable. And if a word has 2 matras or 2 syllables, then you end up with 9 to the power of 2 meanings, friends. So that's why possibly these ancient rishis or scientists chose this language to communicate, because it's very effective. You speak one word, appropriately chosen, one word, and you can create any number of new words if you want to. It doesn't limit you from, saying, oh, no, this is not there in Sanskrit. No, you can choose, you can create, because, you can create new words as well, because the rules are already in place. If you have rules in place, then, playing with those rules is really, really interesting. So the one word, if it has 2 syllables, will have 81 meanings, friends. Why did we lose so much of information? I assume is that we're trying to have a uni-point variate analysis. We are taking one variable and then saying that, it should be equal to this meaning in English, and we lost the rest of the 80 meanings, friends, when a word is consisting of about, 2 syllables. So I applied the principles, of these grammars. I don't know all the 9 grammars, but I know to a great extent 4 grammars: Indram, the Aindram, the Saraswatam, Kashakrutsnam, and Paniniyam. Today, Paniniyam only is vogue, friends, is in vogue. And Paniniyam, the language is guided by about 3,996 grammar principles. Saraswatam is guided by 90,000 grammar principles. Kashakrutsnam is guided by 1,700 grammar principles. And if you want to understand science, this particular secular science we're talking about from our scriptures, you need to know Kashakrutsnam, because, several of these, sutrams I applied to understand these words. And so understanding Chalatva. So, okay, Chalatva, the word that I mentioned here, is translated as mobile. So what most of the Sanskritologists think that Vata, because it is attributed by a property called Chalatva, it moves things. Yeah, while it is right, it also means from Kashakrutsna grammar, but it means mobile when you apply the principles of Paniniyam, but you apply the principles of Saraswatam, Aindram, and Kashakrutsnam. When I applied the principles of Kashakrutsnam, Chalatva also means hyperexcitability. And this hyperexcitability is the one that we proved in this experiment, friends. Why is it hyperexcitable? Because the difference between resting and the threshold membrane is very narrow, and the tissues are they require only very less external stimulus to elicit an action potential and then send a response to the brain. So the tissues are hyperexcitable. Very interesting, isn't it? The kapha is, is characterized by a larger gap between the threshold and the resting. And, the word that is used is sthira. See, look how beautifully the properties are also calibrated. There is a very great sense of quantification of these doshas in the properties itself, which means sthira. Sthira, the kapha systems are sthira or stable. Why? Because the difference between the resting and membrane is very wide, and they require larger external stimuli to get elicited, to elicit action potential, friends. So that's how I started understanding. But then why did the pitta systems elicit almost equivalent response than of the control group of rats? We didn't understand and so we went on with experiments, using acetylcholine and adrenaline on vata, pitta, kapha systems. And what interestingly we found was, while acetylcholine and adrenaline caused expected results in vata and kapha, they had least or insignificant results in effect on pitta systems, which means neither adrenaline nor acetylcholine. Acetylcholine is a parasympathetic mediator, isn't it? Adrenaline is a sympathetic mediator. The sympathetic mediator should have increased the heart rate in pitta diet-fed hearts. Acetylcholine should have reduced the heart rate. But it didn't do any of this. Neither of them could do any of the effect that they otherwise would have produced in normal or vata or kapha systems. But what we found interestingly was the pitta systems had intracellularly, we were looking for other systems, other mechanisms through which action potential is elicited. And we found, in the ancient, again, in the 1960s literature from the Russian, archives, we found an interesting mediator which is called as the alpha-ketogluteric acid, which also is responsible for eliciting action potentials, friend. And where does alpha-ketogluteric acid come from? Comes from the mitochondria. And what we found was, in pitta systems, the alpha-ketogluteric acid is 5 to 7 times more than in vata and kapha systems, friends. 5 times more than in vata and 7 times more than in kapha systems. And we quickly could relate this to, it was found in excessive quantities in cytoplasm. And where could the leakage have come from? It's when there is too much of alpha-ketogluteric acid produced in the Krebs cycle inside the mitochondria, it could have possibly leaked out into the cytoplasm. And we were able to measure this, alpha-ketogluteric acid inside the cytosol, friends. And so the pitta systems had excessive mitochondrial activity producing alpha-ketogluteric acid. And this alpha-ketogluteric acid is responsible for its normal excitation potential. Very, very interesting. It turns out very interesting. What it means in its summary is that vata produces too many action potentials and hence spends ATP faster and hence requires higher levels of glucose, isn't it? Where does ATP come from? ATP comes from mitochondria. Now, the glucose has to be, we all know it's only the glucose molecule that produces ATP, none of the molecules. If you give someone fructose, the body, converts fructose into glucose in the liver, and then, then the glucose enters the cytoplasm and then, gets subjected to glycolysis, then Krebs cycle, then the ETS produces ATP, which means vata requires more ATP and hence the glucose demand is high, and possibly because of it, the glucagon is higher in such individuals. This is one correlation that I want to leave here, number one. I mean, there are other ramifications, but I'll not go into, intra-arterial pressures and all, because it's going to be very complicated. But I limit my discussion only to diabetes today. And glucose metabolism and different possible, glucose metabolic reactions in different individuals, possibly because of this dosha influence. So now look at what is pitta aggravated diet doing, or pitta aggravated diet fed rats or hearts are happening. What is happening in them? Pitta is correlated to mitochondrial activity. When there is too much of mitochondrial activity, what is happening? The body is burning up glucose faster, producing more ATP, but at the end, it requires more glucose, isn't it, friends? See, look, vata and pitta systems have different mechanisms leading to higher demands of glucose, friends. And so in such individuals, if you try to reduce glucose, blood glucose, the tissues are going to subject to, ketogenesis, isn't it? Where does glucose come from when there's higher metabolic activity and blood glucose is lowered because of some aggressive blood glucose lowering therapies? What will happen? The body then mobilizes. It first targets the polysaccharide, storage, glucose storage, in the body, depletes them. Then glycogen storage gets depleted. Then it attacks the fat tissue through a mechanism called as gluconeogenesis. The triacylglycerols and TGLs are all mobilized from the adipose fat, and then blood raises not only TGL but also mobilizes. Acylation happens very quickly, and then leads to production of glucose from new molecules, glucose, through a process called gluconeogenesis, in which you end up producing not only glucose but also ketones. All right. And then when the body is depleted of adipose, the body starts breaking down your proteins, and then leads to production of glucose. Friends, pitta and vata require higher levels of glucose levels. Now kapha, what kapha is doing, kapha as a dosha is doing, is that kapha is actually increasing the resting membrane potential and decreasing it further, widening the gap between the resting and the threshold membrane potential, making the tissues sluggish. If anyone of you have read the guidance physiology of 16th edition, I suppose, the page number 578 or so, it mentions a certain syndrome called lazy leukocyte syndrome. Diabetes is actually characterized by a certain syndrome called lazy leukocyte. Why is the leukocyte lazy, friends? Because of the widened gap between the resting and the threshold membrane potential. These cells wouldn't respond for a normal stimuli, isn't it? So they are sluggish. They're called, this possibly has already been known, has already possibly been aware. These ancient scientists must have been aware, and they carefully mentioned that kapha is attributed by a property called manda. Manda means sluggishness. So friends, what is happening is when the tissues are in a state of hyporeactivity, I call it a hyporeactivity because the reactivity of these kapha tissues is lesser than that of pitta and also vata and also control group of rats. So they are less reactive, and then they use up ATP lesser, and possibly because of which their energy dependence is less, and hence the glucose that you give into the tissues is not utilized. And what happens when you give more glucose to such tissues? End up with glucose produces more fatty acids. All right, the adipose tissue goes higher, and this possibly should be true diabetes, friends. So what I'm trying to again summarize here in this particular slide is that continuing the discussion from the earlier slide, the ATP consumed is very high in vata. Look at the 1, 2, 3, 4, 5, 6, 7th row, friends. The ATP, VED, is for vata enhanced diet fed rat groups. The ATP used up is 5 times more than the kapha. The pitta, the ATP used is only twice, while ATP is generated at a very aggressive very aggressively in pitta systems. Look at the next slide, and then next row. The vata systems use up glucose. The glucose demand is high, but they don't produce, vata systems don't produce glucose. They are dependent on pitta or the mitochondrial systems for production of glucose, and hence the glucose demand is twofold higher than kapha systems. So the pittas, because their mitochondrial, I'm assuming this, I'm not, we cannot conclusively make a claim, but at least, what should be definitely claimed is that the pitta systems have very high glucose. How high? I'm assuming it should be 5 times higher than the kapha systems, because we found alpha ketogluteric acid 5 times in higher concentrations than the kapha systems, 5 to 7 times, as I said. So it should be the ATP glucose demand should be 5 times higher, number one. But the ATP consumed, because they are, the action potentials produced in pitta aggravated diet fed rats, is almost similar to the control group of rats that produce the same action potentials. Their glucose demand is high, while the ATP produced is moderate. So look at the diet, so that is how Where is this source of blood glucose coming in? In Vata, it is diet and acylation. In Pitta, it is diet and gluconeogenesis. And in Kapha, aggravated diet and the glucose storage. What is happening when you give excessive glucose? The glucose transforms itself into either glycogen or, into fatty tissues and so on and so forth. Glucose gets into storage, and such people, become really obese. So, I wouldn't completely claim that it is solved, but a solution looks possible. Current problems with modern medicines is possible because if you understand the Vata, Pitta, Kapha, the glucose metabolism in the light of Vata dominance, Pitta dominance, and Kapha dominance, all the associations, guidelines that are being given by different associations make really sense. American College of Physicians, when they are saying that their people were doing good with 7 to 8 HbA1cs maintained at between 7 and 8, they must have been dealing with Vata dominant individuals. They must have been, I don't know, but we have to again go back to them, retroanalyze, and then come back with their phenotypes and dosha analytics, and then be able to establish this. Because Vata requires higher, Vata produces more action potentials, its glucose demand is higher, and hence the body maintains a certain higher levels of glucose, possibly under the influence of glucagon, and the HbA1c is around 7.10, and they do good. What happens if you maintain lesser levels of blood glucose levels in such individuals? Vata triggers gluconeogenesis, and then depletes not only your fats, but also it leads to muscle wastage. And when the muscle and the fat get depleted, ATP, essentially the ATP that is produced in Vata individuals is very less. So it possibly could have led to decreased contractility of the heart muscle, and must have led to 30. As we mentioned earlier, that people who have maintained their blood glucose levels less than 6.5 in that particular trial called Accord, they 33% of them ended with a fatal heart attack. Why? Because the contractility must have come down, because Vata has used up all the ATP, and the heart muscle must have left with less ATP for its contractility or inotropic effect. So American Geriatric Association must have been dealing with Pitta and Vata people, and hence they feel very good between 7 and 7.5. One of the trials, BADT, Veterans of a Diabetic Trial, really claims this. It says that their veterans were actually, maintaining, when they maintained their blood glucose levels, HbA1c at 9.5, they were doing very well. So American Diabetic Association must have been dealing with Pitta Kapha people, and endocrinologists must have been dealing with Kapha people, which is, I say, is the true form of diabetes trend. And the cholesterol we'll talk about later. So we, applying all this, we came up with our own methodology. The salient features of polyscientific Ayurvedic remedies are, treatment methodologies are, we never try to violate any guidelines issued by scientific congresses, which means we strictly follow the guidelines issued by ICMR. We should never, ever, friends, violate any of the standard scientific congresses. We are not lame people. Friends, we're all scientific people. We have to go only by science. And not only our modern science says that there are 3 types of evidences. There's a direct evidence, like experimental evidence, there's an analytical inferential evidence, and there is a citations, right? 3 evidences that we today look to, isn't it? You know what? Interestingly, there are 3 evidences mentioned in Ayurveda also. There's a Pratyaksha Pramana or experimental evidence, Anumana Pramana or inferential evidence, and there is 3rd one, which is called the Shabda Pramana. In Shabda Pramana, there are citations and also Aptavakya, scientists, great, of the highest orders, words should be taken seriously, friends, is what our science has already been saying. Not only this, our Jagadguru Adi Shankaracharya, he clearly mentioned, Tasmat Shastram Pramanante Karya Akarya Vyavasthito. He says, and hence only scientific pursuit should guide your actions or inactions. What you do should be guided by a scientific evidence. What you don't do, if someone says, no, I don't want to do this, you should ask. Our scientists are not asking. Our spiritual leader, like Jagadguru Adi Shankaracharya, is asking. Do we find such spiritual leaders, friends, today? No. If we don't find them, they're not spiritual leaders. So, friends, let's not get into that discussion at all. Our job is to promote scientific fervor only. And not only did our current modern science is saying, but also our rishis of the highest order scientists are saying that follow only scientific evidence, to take your science further. So what we do is we never violate any of the scientific guidelines. If a patient comes who fits into the inclusion criteria of a certain treatment methodology, we tell them, we send these patients back to their physicians. We say, go back to your physicians, get the treatment done. And we also talk to their physicians. We tell them, sir, your patient has come. If you want us, we can work together, or else you deal with them. If you're unable to treat, because there are several exclusion criteria, or if your patient falls under exclusion criteria, then send them back to us. And by doing this, friends, we made several friends with we became friends with several allopathic physicians by doing this. Several cardiologists, oncosurgeons, diabetologists work with us today, because we never violated any scientific guidelines, friends, issued by the scientific congresses. And we only target end-stage diseases or functional cases, collaborate with the patient's GPs, as I just mentioned. And there's an orientation session, because, you see, this is a new methodology for all practical purposes. The patient needs to understand what we are doing. And then he needs to understand why certain supplements have to be given in a certain extraction format and how they have to be taken, what is the diet that has to be followed. It's not specific diet for every individual. It's a different diet for different individuals. And there's a different fitness regimen, friends. You cannot just blanket, do all the yogasanas. It will not only is it counterproductive, but also it could be fatal. Friends, I'm telling you, there is evidence, of certain yoga gurus dying of a heart attack while performing a yogasana. And those yoga gurus, we cannot call them as gurus, because gurus is a very big word. You can call them yoga acharyas or yoga masters, who have been doing for the past 20 years. He died, one of the yoga gurus from Karnataka, he died of a heart attack while performing yogasana. And he was a master for 27 years teaching yoga. Why? Because every asana has a certain VPK fingerprint. And if you don't understand the original VPK fingerprint of your body at that particular point of time, any asana could do any, deleterious, could cause any deleterious effect. Friends. So what we do is we based on the VPK fingerprint that is expected out of yogasana, we give those yogasanas, workout regimen, breathing exercises, several home remedies, so that, we essentially treat bring about a VPK balance. And then the VPK balance or imbalance is diagnosed through Nadi Pariksha, friends. It's a very advanced science, friends. It's not simple. It's a very advanced technique. I'll talk about it, in maybe during the course later, because it's not in the scope. I have very few minutes left. I don't want to, bother you. But then there's an important take home, is how do we personalize the pharmaceutical drugs is what I need to tell you. I'll take it from here very quickly. So how do we treat diabetes? We assess the patient's condition, the VPK fingerprint of an individual through the Nadi Pariksha, and then, follow all of the salient features of polyscientific Ayurvedic diagnosis, treatment methodology. So you look at this, there are certain anti-diabetic drugs that are classified and, mechanism of actions listed here. So we know that there are insulins, GLP agonists, amylin analogs, biguanides. Could they be personalized, friends? Let's see. See, look at the first classification. I want your attention, please, here. It's going to be very, very interesting, friends. See, insulins. If there is kapha reactivity in an individual, say, which we just mentioned, that kapha makes the tissue sluggish. All right. When the tissue is sluggish, okay, the question that I would ask in and also answer it is, when the when the digestive system metabolizes the polysaccharides or carbs that we take, and the end products of glucose, galactose, lactose, sucrose, several trioses, tetroses, pentoses, hexoses, septoses are produced in the digestive tract. They're absorbed into the blood, right? The glucose molecule that entered into the blood, what is that system that signals that an amount of blood glucose is available in the, what is the, how does the pancreas know what amount of insulin to be secreted? Friends, it is the immune system. The monocyte and macrophage cell lines are the ones that actually act as continuous blood biochemical monitors to actually measure the blood glucose levels and send a signal in the form of interleukin-10 to the pancreas, and the pancreas then produces insulin, friends. Now, at this juncture, if there is a kapha reactivity in the monocytes, what happens? What happens when there is kapha reactivity? The monocyte is sluggish. When the monocyte is sluggish, it doesn't respond to normal concentration of blood glucose and waits until the blood glucose levels go higher, and then the monocyte produces a signal, chemical messenger, and then inform the pancreas to produce insulin. So your blood glucose in a kapha reactivity could be because of increase in kapha reactivity, and blood glucose levels could go up. And if you're giving insulin in such individuals, you're actually causing weight gain, friends. Why? The glucose levels, because there's already kapha reactivity, which is actually predisposing, and we are thinking that insulin is causing weight gain. Look at the possible side effects, and not possibly because of insulins per se, it could be because of the kapha reactivity. Friends, we have to do a lot of research on this. I'm just giving you a hypothesis. But this hypothesis is tested in several individuals. So that's why I'm trying to tell you how we actually don't actually stop all the drugs, but we actually use certain of these techniques in understanding which drug to be prescribed in each individual. So pancreatic pancreatic cells, or if monocytes are in kapha reactivity, then the target should be to reduce kapha reactivity, and early insulin therapy would prove to further augment kapha reactivity, and so should be used when there is a primary deficiency of insulin production. How do we know? We have to do assays. Now let's go to the next. I'm going quickly to each of these classes so that you understand, so that I try to share my mind with you all. So GLP-1 agonist, friends. So what are the GLP-1 agonists doing? They're directly stimulating the GLP-1 receptors. What does it do? It actually increases the insulin release and reduce glucagon release responses. So we all feel at PSA institution, the GLP receptor agonists are well suited for early use in type 2 diabetes because they stimulate release of insulin and suppress glucagon secretion only when blood glucose concentrations are elevated, friends. Thus, the risk of hypoglycemia, while it is low, they are better suited for kapha reactivity of pancreas and monocytes, friends. Now, the amylin analogs. When there is already, for example, if you have a patient who has an individual who has pitta reactivity, we just mentioned that the pitta reactivity is actually leading to hypermetabolism. All right, this hypermetabolism is increasing the demand for glucose because the body is burning up glucose faster. Now, in pitta reactivity, the body increases the threshold of blood glucose burns faster, and the body increases the threshold of blood glucose levels by increasing the glucagon production so as to compensate for the excessive mitochondrial activity, and possibly thyroid possibly acts through this mechanism, and we cannot of course equate pitta reactivity with thyroid excesses, but I'm just giving an example. In excess thyroid hormone increase, excess thyroid hormone also increases lipolysis and secretion of both insulin and glucagon, friends. And the sum of these effects deteriorates glucose metabolism, which can cause glucose intolerance and diabetes in pitta reactivity. So amylin analogs are good possibly only for pitta reactivity, friends. Now look at the biguanides. What are the biguanides doing? The biguanides at the molecular level, metformin, for example, inhibits the mitochondrial respiratory chain in the liver. All right, inhibit. So, as we said, in pitta, the mitochondrial activity is very high. All right. So, because at a molecular level, biguanides are actually inhibiting the mitochondrial respiratory chain and leading to activation of AMPK, enhancing insulin sensitivity, via effects on the fat metabolism and lowering CAMP. All right, it reduces the expression of gluconeogenetic enzymes, and hence the biguanides could be very useful in pitta aggravation and not in vata or kapha. All right, sulfonylureas, and even the next class, meglitides, what they are doing is directly stimulating the release of insulin from pancreatic beta cells and thereby lowering blood glucose concentration strength. Because they work by stimulating insulin secretion, they're useful only in patients with some beta cell function, again to be chosen in kapha reactivity of pancreas and and the monocytes, friends. Now, DPP-4 inhibitors, the classical mechanism says that they inhibit the DPP-4 activity in peripheral plasma, which then prevents the inactivation of the incretin hormone glucagon like peptide GLP-1 and in the peripheral circulation. And DPP-4 inhibitors thus lower blood glucose by helping the body increase the level of the hormone insulin after the meals, friends. So insulin, it helps to move sugar from blood into the tissues. So, it should be chosen only in vata reactivity, friends. You see the point, right? The point I'm trying to make here. Now, the SGLT2 inhibitors, what are they doing? They function through a novel mechanism of reducing renal tubular glucose reabsorption. Friends, I'll share something really interesting. Several Pitta individuals in our patient category and Vata individuals. So as I said, Pitta and Vata, they increase the threshold of blood glucose. They don't actually it shouldn't be treated as diabetes. It should be treated as reactive and compensatory hyperglycemia. Why reactive is because the body requires higher levels of blood glucose so as to compensate for the mitochondrial excess that is being caused because of pitta aggravation in pitta aggravated individuals. In Vata, because of hyperexcitability, the body requires more ATP and hence the body requires more glucose, friends. So this is compensatory and reactive, respective compensatory and reactive, glucose responses, and hence the body, what it does, if there is excessive glucose in the blood, what will the body do? The body will push it through urine, isn't it? But several of these individuals who have pitta and vata aggravation, friends, we found that they don't have glucosuria. They don't have blood glucosuria. So glucosuria possibly should be a symptom only in kapha reactive individuals and should be SGLT2 receptors, appropriately should be chosen in kapha reactivity. And hence, alpha glucosidase inhibitor should be chosen in kapha reactivity. Thiazidine diazones, again, diones should be chosen only in vata pitta reactivity, friends. I'm trying to cut short, because I don't want to, quickly wind up. Yeah, sure. We are winding it up. Just give me a moment. So friends, several people were treated, including IDDM patients, several patients were treated. And, I'll show you a case of IDDM patient who was a 4-year-old patient who was just 12 kilo in weight, and with anti-IA2 receptors, 2000, it should be less than 10 at the beginning of the treatment. And you know what? He was identified as a vata excessive individual. We loaded this patient with both excessive glucose and also excessive fats. What happened? After 2 years, look at this, the anti-GAD antibodies came down to 9.64, and the anti-IA2 insulin, which was 2000, came down to 195, and patients' insulin levels were, insulin prescription were reduced by at least 10-fold. Several patients we treated, several case studies that I can present, not just in diabetes, but also, in heart diseases were treated with this. There's a vata-mediated heart disease, pitta-mediated, kapha-mediated heart disease. We treated by appropriate remedies, friends, and psoriasis, several diseases, friends. Look at this kind of results you don't get anywhere. We target only end-stage patients. We treated several diseases with allergies, asthmas, diabetes, autoimmune disorders, several autism patients, senile dementia patients, chronic infections, friends, in the last 20 years. With amazing success. And, we identified more than 3,000 markers for Vata Pitta Kapha quantification. And we were the first in the world to demonstrate complete regeneration of the heart and other organs in vivo. When we did, when we induced MI, we demonstrated how MI could be mitigated, not MI could be mitigated. Post-MI scenario, we were able to demonstrate total regeneration of the heart through this technique. We demonstrated how brain can regenerate, the pancreas can regenerate, the kidneys. Not totally, because in partial nephrectomy models only we were able to demonstrate the liver, subtotal hepatectomy models, we demonstrated how livers could regenerate, friends. It's a very, very, very interesting, subject that we have come up with. What I'm saying is this polyscientific Ayurveda has helped us take allopathy 10 steps ahead, and we came up with a new device for this as well, which this device could actually measure your VPK individual fingerprint, and then prescribe personalized dietary herbal supplements, physical exercise, yoga regimen, breathing exercise, and home remedies. Friends, this coming very soon into the markets. It's not about that. There's not a marketing gimmick, but I'm just sharing this information. And several markers, as I mentioned, were identified, and we, based on these markers, we actually quantify the VPK imbalance in an individual. And I definitely see that we can really position Bharat as a health capital of the world if this science is taken forward into a tertiary model, in which the tertiary model of hospital could be supplied with several polyclinics. With this device, we can automate the working of all these polyclinics, which had as feeders to this tertiary hospital, and tertiary hospital could deal with several end stage diseases. And then we could actually take this to treat several functional diseases and also end-stage diseases. That itself is a very huge market, friends. $1.2 trillion is spent in the world. That additional 1.2 trillion revenues can come to India. This is what the point that I'm making. And also farmers could get benefited. I frankly believe that if Americans are sending rovers to Mars, and if we send Mangalyaan to Mars, it doesn't make India great. No, I feel that we have to put our own sciences into action, and then empower different fields of our health. I mean, as doctors and scientists, we could actually empower, improve the health of our populations through our own indigenous sciences, and that's the way we could actually help India become a health capital of the world. And applying different subjects by doing appropriate research, we could different ancient sciences, we could actually apply that into different aspects of our social living, and then make India the health capital of the world by translating our own indigenous sciences into bringing our own indigenous sciences into a certain methodology and empowering India, and make India the health capital of the world by in Indian way, by saving money or saving lives, friends. This is what I frankly feel that we can really make India the health capital of the world. And friends, take home messages: India is a cradle of human civilization, endowed with amazing indigenous sciences. Friends, we don't become superpowers by copying someone else. We become superpowers by applying these sciences to solve current problems of the world. And thank you very much, Mujib, and all the listeners here for giving me this opportunity to present it. And thank you very much. I'll take the questions. There is some time left. I ran over.
Thank you, Ravi Shankar sir, for making this session in a super good way. So Divya, we are open for questions. If anyone have any questions or any kind of things to be discussed with Dr. Ravi Shankar sir, they can type, and you can take it up questions from the chat box. Dr. Divya.
Dr. Venkat Joshi, thank you very much, Ji. Thank you very much for your blessings, sir. In UK, we had a wonderful time with Dr. Joshi, who runs the Ayurvedic Association in UK, along with the British parliamentarians. Dr. Suresh Swarnapuri, thank you very much for your kind wishes, sir. He runs the European Ayurveda Academy, and does some wonderful work there. I was invited as a faculty there very recently to help in their fellowship program, and really very I'm really happy to be associated with them. And then is Dr. Poly device available in the market? It's not yet. It's going to be available as a form of a pre-sale. The offer is going to be very soon let out into the markets in the month of November. Dr. Mujib will share this information very soon. In the market, there's so many drugs, and then why is there diabetes not cured? As I said, Mr. Raju, there are several drugs, but I feel they're not personalized, and the they're not personalized based on individuals, either dosha-based preferences or genomics. So there's a lot of work that could be done, is what I was saying, that as I mentioned, I mean, if you have looked into my PowerPoint in the 47, 48, 49 slides, I clearly gave the list of all the classes of anti-diabetic medications available in the market, but I also mentioned their limitations based on my dosha understanding. So if this VPK understanding is generously spread across, then we could actually personalize these drugs as well and possibly come up with a cure. But as I said, we have more than 4,000 patients whom we dealt with, who suffered from end-stage refractory diabetes, and we were able to help them increase their quality of life and also symptom-free survival, Mr. Raju. Thank you very much. What are the key points to maintain VPK balance? I mean, that is a very tough question because each individual has a specific VPK fingerprint. You cannot have a standard, template for maintaining your VPK balance. I would say that, today, in today's world, because of mixing up of several ethnic populations, the doshas also got mixed up. You don't have that clear-cut vata dominant, pitta dominant, kapha dominant people, as mentioned in the ancient texts. Today we have mixed VPK VPK dominant people, and again, these VPK imbalances are not homogeneous across in an individual. They would have different VPK imbalances in different organs. So detection of these imbalances is very important. And based on which you can personalize recommendations. That's to Mr. Nadav. 1+60. I don't know who's using this. Your name is not visible here. I've said about athiloma, athikesha, naka, and okay. See, these are again. See, explaining vata, pitta, kapha itself took more than an hour. I mean, it's not complete yet. Again, if I have to go to athiloma, athikesha, naka, and all, they will take at least several hours, friends. So that is reserved for a polyscientific Ayurvedic course. I mean, the course is a 545 hours of lecture for everyone. I mean, it is like lecture for both Ayurvedic doctors, allopathic doctors, homeopathic doctors, Unani-based doctors, Avicenna or Chinese medicine-based doctors, because this course will help us interact with each other. We would have developed a certain common language through which we can speak to each other. Because if today if you're talking about athiloma, how would you, even vata, for example, how many allopathic doctors would understand what is vata? They wouldn't understand. Now, if you talk about SLT4 inhibitors, for example, SGLT2 inhibitors, sorry, how would an Ayurvedic doctor understand? So there's no common language, first of all. What I'm trying to say is, let us work together to talk with each other first, because we're not even talking, we're fighting with each other. Allopathic doctors are fighting with the Ayurvedic doctors because they feel that these guys are not scientific. Ayurvedic doctors say that our science is very advanced and you guys are very primitive and we don't want to talk to you. Unfortunately, this this fight is actually diminishing us. I feel through polyscientific Ayurveda, a language is created and can further be improved to make our understanding between allopathic doctors and Ayurvedic doctors better, so that we collaborate with each other, we work with each other and not fight against each other. So that's why, I'll explain in PSA way very soon, but not today because it's already, it needs to be wind up. Thank you, Mr. Rajkumar. Rajkumar ji, start reading with some Vasantlath books, please. That will give you a basic understanding of VPK. Thank you very much, Sapna ji. Ayesha ji, VPK knowledge. Yes, as I said, PSA course is being unraveled very soon. Dr. Mujib and I and several other people from EAA, the European Ayurveda Academy, run by Dr. Suresh Varnapuri, Dr. Joshi ji, we're all working to bring this course live. So that's when, you all will have this VPK knowledge, Ayesha ji. And quantify, yes. Jefton James, personalized treatment, as I said, 63% of our patients are free from all medicines, allopathic and also Ayurvedic. 20%, we are coming with the stats very soon, Mr. James. Dr. James or Mr. James, I wouldn't know. But, in diabetes, as I said, recurrence of diabetes. So this will be discussed in this conference that we are presenting very soon. The venue, the dates will be known, will be made known very soon through Dr. Mujib again. Stay in touch, Mr. James. That's going to be a 2-day session on several diseases, and we'll present more data statistics, and also case presentations that will help you unravel, understanding, this particular science, friends. I'm Pilla Venkatesh Rao from Vijayawada. I've started taking your business. Oh, Mr. Venkatesh Rao Pillaji, thank you very much for coming out open to share your, we didn't call our patients. Please don't feel that it is a marketing stunt at all. It is not at all a stunt. This is a, this is a mission, friends. You and I and everyone around, it is our duty, friends, to take this mission forward, because with just 1/10 or 1/20th of the cost, people really getting better, friends, and it is improving the quality of life and symptom-free survival. So, Venkatesh Pillagaru, thank you very much for sharing this. We're going to present several of our patients during this PSA conference, and help you understand the implications and applications of this particular science. Professor Raghupati ji, I'm really honored to have your presence here, sir, and with your blessings, I'm sure this way we'll take this forward and help our youngsters create and further develop this new science of polyscientific Ayurveda. And from India, I really want Sanmati ji, Sandeep ji, Blessy, Lata. I mean, all the youngsters that I see here, I really get motivated when I see you, friends, and including Mujib. Mujib is a very young person. At heart, he's only 15 or 16-year-old. And with his team, an excellent team he's got. And I'm very hopeful that we all together can actually, bring this, further develop this science of polyscientific Ayurveda, make India the health capital of the world. How? By presenting this knowledge to the world. And it has to go from India. That's exactly why I gave up my residency in Canada and came back to India to, make this happen. And then together we can really make it happen. Pantulu Raghupati Garu, thank you very much. Rajkumarji, thank you very much. If no one has any more questions, we can wind up, Mujibji.
Yes, sir. We'll close this session, but before closing, I think we are planning for one more big conference, which we'll be sharing shortly. And about the device also, we'll be posting in Clinosol Research and SGP, the date and the way we are looking forward to take it ahead. So we'll be sending a feedback form also. Please do complete your feedback form and any questions you have, Dr. Avishankar sir. So, and your certificates will be sent in, I think, 2 or 3 days. So I think that's
Mr.
good,
Verma, I'm sorry, Mujibji, I'm sorry. Mr. Verma says any papers published in cancer and autoimmune models in rats. No, we have tried PSA in end-stage cancers and autoimmune disorders, lupus nephritis, and several other autoimmune diseases, SLEs, ITPs. Multiple sclerosis. We're presenting these cases and also presenting the papers. They're under review right now. They'll be published very soon, and we'll share it, in Clinosol, through Clinosol. We'll share those papers, Mr. Verma. Thank you very much. Carlos, thank you very much, my dear friend. Sorry, Mujib.
So I think we can drop off, sir, and thank you, and happy Diwali to everyone. Thank you, sir. Thank you.
Thank you, Mujib. Thank you, Mujib. Thank you. Happy